I would like to first thank the organizers, Andras, Giorgio, Sofia, and Sofia. Thank you so much for inviting me here for the first time in Hungary. Actually, we have a postdoc from this city, so she gave me lots of information about where to see, where to eat. I don't think I have time to do that, but okay. So I'll be talking about our work at XFEL. I'll be talking about photosynthesis. This was already mentioned in some of the morning talks. We know about photosynthesis, and what I will be talking about is the water oxidation part, which is catalyzed by the protein, membrane protein called photosystem II, which Petra also mentioned. In photosystem II, if we look inside, catalytic center is 4-manganese-1 calcium cluster, which is right here, surrounded by water molecules and amino acids. So we want to see how this catalytic center works. As you probably know very well, this is how the charge separation occurs at photosystem II. Chlorophyll center absorbs red light, and then there are 4 photochemical reaction occurs right here during the water oxidation reaction. So 2 water molecules are spread into 1 molecular oxygen for protons and 4 electrons. That is happening at the metal catalytic center, manganese center, which goes through the 4 redox state changes during this catalysis. So we want to see how this enzyme works, but why? This is my version of why we want to study photosystem II. So half of my life, I'm working on more material science-oriented research, for example, CO2 reduction reaction, which is very difficult if I try to do it based on material sciences. First of all, the selectivity is not good, and at least 2 electron reduction, there is a way to go. But if we want to make a higher C product, it's a very difficult reaction. So we think that how to manage multi-electron, multi-proton reaction, we have a lot to learn from the natural system, particularly natural photosynthetic systems. So this is first motivation. The second motivation is that we want to establish a method to follow structural and chemical changes of biological system, which is by nature, it's a very dilute system. So we are working on crystallography and the X-ray spectroscopy. So decades ago, I think our understanding of the catalytic reaction of water oxidation reaction was something like that. There was a high resolution structure of the resting state at cryogenic temperature. So we knew the starting point, how it looks like at the synchrotron facilities, but we really wanted to know how things changes structurally and chemically. So currently, our understanding was much more than in the past, and I want to talk about how we came to this understanding. Some of them are still hypotheses, and it's not only our group's work, but as a community, I think we gained information a lot. So this is based on the availability of X-ray free electron lasers, method development, and sample improvement, as well as there are use of high-power computing facilities, and all the software has been developed through this process. So that's what I'll be talking about. First, about the sample. We grow this cyanobacteria with bioreactor, and then purify photosystem II, then crystallize it. For some spectroscopy measurement, we use solution sample, PS2 solution samples for crystallography, plus X-ray emission spectroscopy. Our sample is single crystal samples. Then we do serial crystallography or shot-by-shot X-ray spectroscopy like this. When we started, this was a kind of unit cell distribution of photosystem 2, A-axis, C-axis, and the resolution. So there was a quite high heterogeneity. In this case, we can only merge a fraction of data in order to get complete structural information, right? It's not surprising to have this kind of high heterogeneity because if you think about protein crystals, which is more than 60% is water, depending on the crystallography, crystallization condition, the unit cell is slightly different. But 1 angstrom, 2 angstrom differences of the unit cell is really killing us. In the future, I imagine that we can deal with this kind of heterogeneity to merge all the information together instead of throwing out most of the data. So we think we want to go towards that direction. But for now, our effort is how to make as monodisperse crystal unit cell as possible. And that is possible. We did that. And this also improved the diffraction quality to close to 1.9 angstrom because most of the data now we can merge and then create a structure. However, this is an ideal situation. Still, it's really challenging to control unit cell. And sometimes we see this kind of unit cell structure. So there's a secondary unit cell appearing. This is not good. But what I want to say is that we have a control to move this kind of distribution to ideal distribution during the beam time. So that's what we gained. Regarding the access to various time points, this was a topic of this morning's talk as well. In the photosynthetic system like photosystem 2, we want to look at the various time points starting from, let's say, femtosecond, picosecond time scale, which is related to charge separation stabilization, down to the catalytically relevant time scale, which is micro to millisecond time scale after comp laser. So that doesn't mean that we don't need a femtosecond X-ray laser. We still need that in order to avoid the radiation induced changes. So this is a probe pulse is femtosecond. But we have a delta T, long delta T, if we want to look at the structural or chemical changes relevant to the catalytic center. So we have a delta T of microsecond, millisecond time range. So to access various time points, which is quite widely spread, we introduced this sample delivery method. We have we called conveyor belt, which is in which the acoustic transducer is incorporated. That makes a droplet like this. And then we have a 3 fiber coupled optics plus 1 free space pump laser. Combination of these gives us access to various time points, like a stable S-state, as well as time points in between. So we should be able to do the experiments from several hundred femtosecond up to millisecond time scale with this. I'm sure many of you saw this setup in one way or another. So this is our tape drive. This is the transducer, which produces droplet. The diameter is about 150, 200 micrometer diameter. And the droplet goes through this reaction area where we have a fiber coupled laser. And how many flash we give, we can make a S1 state, S2 state, or S3 state. And then we have one more visible laser right here that gives a finite time slice, and then X-ray comes here and then destroy droplet. At the downstream, we collect diffraction, X-ray diffraction for crystallography, and orthogonal direction right here. We have a von Hammer's spectrometer for X-ray emission data collection. This gives us a oxidation state of the manganese cluster. So we can get these two structural data and spectroscopy data simultaneously by shot. This looks nice, no? But in reality, the setup looks like this, and this part is only this part, and the rest is wires and various things, including chemotype. So with this setup, we could go through various states. First, this was almost 5 years ago. We went through the stable S state. So this is the dark stable S1 state, manganese cluster density, S2 state, S3 state, and this is the S0 state. So this flashed state is not necessarily 100% pure state, but we know from other experiments like EPR or membrane mass inlet spectroscopy, we know the distribution of S state, and based on that population, we extract pure S state structures like this. So we see that oxidation state of manganese changes one by one from S1, S2, S3, and then going back to the most reduced S0 state. What we see is in the S3 state, as the resolution started increasing, we started seeing this additional ligand. We think it's OH coming into the open coordination site. So this manganese was 5 coordinated in the S2 state. And going to S3 state, we start seeing this additional density right here. In order to see this density, we needed to reach certain resolution, crystallography resolution. So this is the current S3 state. If we dissect electron density right there, see the plot looks like this. If the resolution is about 2.2, it's hard to see 2 oxygens present here, but reaching 2.0 and better, we start seeing some humps. So this gives us a confidence that there are 2 oxygens right here. Still, I would say getting accurate chemical structure based on these electron density map is challenging. So that's what we are working on right now. But based on the stable S state structure, we started collecting data between the stable S state. This is to see how the structure, one structure evolves to another next state structures. So for example, S2 to S3 state, electron density of what I'm showing here is the oxygen we call O5 in a blue color, and the additional density gaining right here is in the red circle. We see that somewhere around this time point, 150 microseconds after the second flash, we start seeing this density and growing. So this is the OX density. Based on this, we see that when this additional ligand coming into the open coordination site. Then we wanted to look at the last step, which is the most interesting step, where the S3 state goes back to the most reduced S0 state by releasing molecular oxygens and going back to the original state by getting water substrate. So we did a similar experiment. Oh, by the way, the question is, the question we are trying to answer is that which 2 oxygens are coming together to make an O bond, and how the catalytic center goes back to the original state, because I think that is the most difficult step for any catalytic reaction in order to reset and then repeat the reaction again and again and again. This was shown by Petra too. So this is how the electron density of O5 and this newly inserted OX density changes through the S3 to S0 state. So you can see the density changes. Right? During this time, this process, what's happening is that last oxidation of this cluster happens. So one electron needs to go out. That is supposed to be a so-called S4 state. And then right after that, 4 electron needs to be reduced by releasing molecular oxygens and getting water substrate back. We see some electron density shape changes in this area around the OX. So our interpretation is that somewhere around here is what O-bond formation happens. And then slightly later, we start seeing the decrease of OX density, disappearance of OX density. So we think this is the stage that molecular oxygen is released and the catalytic center goes back to the original structure. If this is the case, when O-bond formation happens, this is the first 2 electron reduction step. And then the release of molecular oxygen is the second step of 2 electron reduction. So what we are thinking is that the reset of the catalytic centers occur in 2 steps. Petra mentioned that we don't see 2 oxygens disappearing. Our hypothesis or our interpretation is that the recovery or insertion of water to this original O5 place location is very fast, because probably that's how the catalytic center wants to stabilize its structure rather than keeping the open space for a long period of time. So that's our interpretation. Anyway, so through that experiment, we could take a snapshot of various moments during the catalytic reactions. As I mentioned, we see the changes, but to get the accurate chemical structure, modeling the chemical structure based on this still requires better resolution, and that's what we are working on. Then up to here, I'm only focusing on what's happening at the metal catalytic center, but protein is big, so we also want to see what's happening at the rest of the protein. We see many things. The earliest time point we collected so far is 20 microsecond, if this is the step, 20 microsecond after the third flash. So at this point already charge separation happens, finished. So what we see probably is the consequence of that, and we see the tyrosine residues right here, plus histidine ligand changes, which is located at the P680 and the manganese cluster. And then we see some changes around the quinone QA side. And slightly later, we start seeing some changes at the manganese cluster around 200 to 500 microsecond. And reaction continues. We see some changes at the mobile quinone QB site around 500 microsecond. What's most surprising is that even at the millisecond time scale, like a 2 millisecond or 4 millisecond, there are still some changes continuing. And the step which is taking quite a long time is the rearrangement of the hydrogen bonding network around the channel region or around the manganese cluster. So the reset process of the catalytic center seems to be taking quite a long time. I want to talk about the proton channels. So during this reaction, 4 proton is released. We haven't looked at this one, so I'm focusing on this proton and these 2 protons. So S2 to S3 transition, 1 proton is released. The S3 to S0 transition, 2 protons are released. And question is, which potential these channels are used for the release of protons. So what we see is what we think is that proton is released with this so-called chloride 1 channels, and I will tell you why we think so. First, S2 to S3 transition, where 1 proton is released. So this proton is likely coming from this one. Water ligand is coming into the open coordination site, so likely H2O is becoming OH. So this is the point that 1 proton is released. I told you we started seeing this density around 150 microseconds after the illumination. Similar time point, we see the structural changes around this bottleneck area where the channel is very narrow. So one of the ligand, glutamate 65, is rotated around 52 rotation. And then by 250 microseconds, it goes back. So there's a reversible motion. So we think this is the timing of the release of this proton through these channels. If this is only the observation, we are not sure is this related to the insertion of water or release of protons. But the next observation gave us more confident. So we looked at the S3 to S0 transition. This step, 2 protons are released. First event right here, I think, is when the 2 oxygens are coming together. So this one is O, this is OH. So 1 proton needs to be released at this point. And then the second release of proton is when this empty O5 position is filled by water molecules. Again, H2O needs to go back to OH. So this is the release of second proton. Interestingly, we see the structural changes of same residue, glutamate 65, earlier time point around 500 microseconds, and then it has a reverse motion. And then once again, it moves around the later time point, 1200 microseconds, and then get the reverse motion. So since the same residues moves twice, it looks like maybe this channel is again used for the release of protons. So this is our interpretation. Okay, so to summarize the outcome, we could visualize structural and the chemical structural changes under functional condition with X-ray method, crystallography, and spectroscopy. And what we are learning is that role of protein environment during the multi-electron, multi-proton reaction is very important. So we need an environment, we need a protein. We are doing one more experiment. So what I've been talking about is crystallography plus X-ray emission spectroscopy, right? We are doing X-ray absorption spectroscopy as well. This is to look at the Zenith region or X-axis region to see how the oxidation state of manganese cluster changes or the structure of the manganese cluster changes from the X-axis region. So, I mean, X-ray absorption spectroscopy has been widely used at the synchrotron facilities, but at the free electron laser, this is a little bit tricky because we need to monochromatize the beam. So the intensity of the incoming X-ray fluctuate almost 0% to 100%. So that's a quite inefficient experiment. If we do the X-ray absorption measurement like a traditional way, it looks like this. We have an incoming X-ray, it hit the sample, and then we are collecting fluorescence, manganese fluorescence signal from the sample to the detector. If we do that, we get the data like this. There is a shape of manganese KH, but it's quite noisy. That is because we don't have a way to discriminate the other photons on top of manganese fluorescence photons. So the method we decided to take is to add analyzer crystals between the sample and the detector. By this way, we use the black reflection from this analyzer crystals and then get only manganese fluorescence data into the detector. So compared to this situation, the background is very quiet. With this method, we could collect the data like this. So this took 2 hours. This took 20 minutes. That tells us that we really need to take care of background noise for spectroscopy, which we know, but nice to see it. Okay, this is my summary. I already talked about how we are approaching visualizing structure and chemical sequence of event, and simultaneous data collection of crystallography and spectroscopy helps understanding the relation of metal chemistry plus protein environment changes. So light acceleration, like what photosystem 2 does, it's relatively easy to access because we can clearly illuminate the samples. But this is just a fraction of the samples. Others are like chemically induced changes with gas, like oxygen, or substrate with solution. So we've been using same setup, but with different modules to access these changes. With that, I want to thank all my collaborators and co-workers. One more slide, and also the facility. And thank you so much for your attention.
Thank you very much, Junko. Any questions? Yes.
Really awesome talk. I have a question about what you mentioned at the beginning, that you will try to merge the data even if the unit cells are very different. So what is, you are developing new algorithms to do that, or?
Yeah, it will be nice to go towards that way, right? I think that's where AI/ML approach should help to scale different data into the same data. I believe that's what I naively think about it. But in both cryo-EM and the X-ray, X-ray crystallography, we have so much dark data, but I think we should be able to use all these data.